Okay, we'll watch this video here on translation, which is the processing of the mRNA molecule to the polypeptide, which will become a protein. So translation occurs out here in the cytoplasm.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into three stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated.
Just a couple of comments here. Our textbook doesn't mention the E site, so you don't have to worry about that. And don't worry about the elongation from the 5' to the 3' area, 5' to the 3' direction. Again, don't worry about those details. But you do have to know the P site and then the A site. If you think about it, the P site is where the polypeptide chain is being elongated, and the A site is where the tRNA is bringing in a new amino acid.
Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
All right, so let's look at the elongation process. Amino acids are added one by one in a three-step elongation process. When step one is codon recognition, and that's here, that's where the anticodon on the tRNA pairs with the codon on the mRNA molecule, and that tRNA will come into the A site. Then, in step two of elongation, peptide bond is formed between the existing polypeptide and the new amino acid brought in. As it shows here, the little red line is indicating the new peptide bond. And then the ribosome functions to create the peptide bond. And then in step 3, translocation, the P site tRNA leaves the ribosome. Being ejected, it's already transferred its amino acid to the growing polypeptide chain. And then the tRNA that was in the A site shifts to the P site, and in doing so the mRNA molecule also shifts across. so that now the A site is now vacant and open for the next tRNA molecule to come in, bringing in the next amino acid. All right. And then finally, the terminations phase. A stop codon should be one here. Here we go. Termination. The stop codon of the mRNA moves into the A site, and the completed polypeptide chain is then released, and the ribosomal unit breaks into its small and large subunits, and the mRNA molecule is released. And then the polypeptide then gets modified, gets its correct three-dimensional shape or configuration, and is sent off to wherever it's supposed to go. So a quick review here. From DNA, the gene or the allele, to the RNA, mRNA molecule, once it leaves the nucleus, to the polypeptide that will become the functional protein. So this is the flow of genetic information in the cell from DNA to RNA to protein. And again, the protein is going to have some characteristic or trait. So we're really talking about the genotype to the phenotype. So in transcription, the DNA, a section of it, the gene or allele, gets copied and becomes the RNA molecule. It gets processed, and then the mRNA molecule leaves the nucleus, going out into the cytoplasm, where translation occurs. The mRNA molecule latches onto the ribosome, and tRNA molecules bring in the appropriate amino acids, forming the polypeptide chain, and then the protein is ultimately produced. And it's a rapid process. Single ribosome can make an average size protein, 400 to 600 amino acids in length, in just under a minute, in less than a minute. So transcription and translation are how the genes, alleles, control the structures and activity of the cells, right? How the genotype becomes the physical phenotype. So this flow of information originates with the specific sequence of nucleotides in a gene or allele, right? On the entire DNA molecule, only a certain region of it, the gene/allele, gets expressed to produce the gene product, the protein. And then proteins that are the final product of the polypeptide, once it's modified, determines the appearance and capabilities of the cell or the organism, its phenotype. So a little video here wrapping most of this material up for protein synthesis. And we do have plenty of time here.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
And that enzyme is RNA polymerase.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. the small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid.
Right? That first tRNA comes into and binds to the start codon. And then the next tRNA comes into the A site.
The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
So, nice little review there of protein synthesis via transcription and translation. Okay, so mutations do occur, and a mutation is any change in a nucleotide sequence within the DNA of a cell, and that is a mutation. So, mutations can involve large regions, meaning a number of nucleotides, or just a single nucleotide pair. A single nucleotide mutation, what's called a point mutation, is the cause of sickle cell disease. So the misshapen hemoglobin molecules as a result of this single nucleotide base pair error or mistake. So if you see here, the normal hemoglobin DNA gene or allele is CTT. When that's transcribed to the mRNA molecule, it should be GAA. And when that's translated to the appropriate amino acid, it should be glutamate. But with the mutation of an A nucleotide instead of the T, that becomes the U base on the transcribed mRNA molecule. And when that's translated, notice that it causes a change in one amino acid in the now sickle cell hemoglobin. And that's enough, that one point mutation, single nucleotide, is enough to cause this disease. Just one. Types of mutations. Mutations of genes can be divided into 2 basic categories. Nucleotide substitutions, like what we saw with sickle cell there, and nucleotide deletions or insertions, meaning exactly what the term means. Nucleotide is inadvertently lost, deleted, or nucleotide is inadvertently inserted, added to the original sequence. So missense mutations are due to that single nucleotide substitution, and that usually can cause a change in the amino acid coding, like we saw with the sickle cell hemoglobin molecule. Nonsense mutations, an amino acid codon is converted into a stop codon, so the production of the polypeptide is inadvertently stopped before it should be, with a nonsense mutation. And then mutations involving the deletions or insertions of one or more nucleotides usually cause what's called a frame shift mutation, because everything after that has been moved out of the original sequence. And that usually causes a multitude of incorrect amino acids to be brought in, and that's disastrous.